Good morning, everybody, and welcome to Global Neuroscience Institute Grand Rounds. And now it is my great pleasure to introduce our speaker, Dr. David Irwin. Dr. David Irwin directs the Penn Frontotemporal Degeneration Center at the University of Pennsylvania Perelman School of Medicine and is the principal investigator of the Penn Digital Neuropathology Lab. He has dual training in cognitive neurology and neuropathology, and his lab focuses on integrating human brain histopathology and molecular techniques with imaging methods to discover therapeutic targets and develop tissue-sensitive biomarkers to facilitate clinical trials for emerging therapies for frontotemporal dementia, Lewy body disorders, and Alzheimer's disease and related disorders. Dr. David Irwin will be speaking today about translational medicine advances in frontotemporal dementia. Dr. Irwin.
Thank you, Dr. Gleavis, for the wonderful introduction and invitation to speak to you all today. I'll be sharing some exciting research that we've been doing and giving an overview of frontotemporal dementia. I have disclosures and funding sources, and I wanted to start the talk today going back to when I started my training in cognitive neurology. At the time, there was a clinical trial for memantine being done for FTD, or frontotemporal dementia, and it was an exciting time. I had learned about this in training and in residency about, relatively uncommon dementia that's characterized by various language and behavioral features that we'll talk about this morning. And the fact that an international clinical trial was being performed was really exciting. It was something, just the performance of a trial was a major accomplishment. And I highlight here a editorial that was done on the paper publishing the results of that clinical trial. And unfortunately, that trial failed to reach its endpoints. And importantly, memantine was found to have mild but statistically robust associations with adverse outcomes in FTD in terms of cognitive function and behavioral features, suggesting that memantine is not appropriate for use with patients with FTD. And while this outcome was disappointing, it was important and an important step forward. And one of the core takeaways of that study that's highlighted here in the editorial, titled Hope Abandoned, the failure of memantine therapy to reach endpoints in FTD, is the heterogeneity of the disease. So one of the topics I'll be going through is how clinically diverse these disorders are, and similarly pathologically how diverse the biologies are. But we're learning a lot and come a long way since that time. And so the overall outline of the talk will be in three parts. One is a discussion of the approach. One of the difficulties with this disease is that we don't know what's happening biologically in the brain of a living patient that we see with FTDs. So we have a robust brain donation program for individuals who participate in research during life, where we could start to put together the pieces of how to improve our diagnosis as early as possible. And then the application of these methods of studying the brain that we do in my lab to help model the microscopic disease. As you all know, in neurology, model systems are challenging for the human brain. No, organ system is probably as challenging to model than the human brain. So using human brain data itself to guide us and help us model what's happening in the disease is important, and show some of what we've learned from that. And then the translation, where we are now with clinical trials in FTD. We have several active trials here at Penn that are based off of the type of observational research that I'll be describing. So some background information on the importance of FTD, although it is a less common dementia in the young onset range, which is somewhat arbitrarily defined as less than 65, or most prominent between the ages of 45 and 65, the prevalence is as equal as Alzheimer's disease, and it's about 20 per 100,000 people. And what's unique of the challenges of the care for these patients is that as opposed to a later onset dementia, patients are usually active in the workforce and have dependent children at the time of diagnosis. That can put caregivers in an extremely difficult situation where not only they have an otherwise healthy adult who has cognitive, behavioral, motor, and or language difficulties, but also dependent children, and could be very difficult to manage. And studies that formally look at the economical and personal impact of a dementia in this age range find that these costs, both to individuals and to society, exceed those of a later onset dementia. So young onset dementia and FTD is an important public health problem. We lack FDA approved therapies. So we often borrow from psychiatry, symptomatic medicines that can help manage behavioral symptoms, and supportive care is the core of therapy. We need new therapies, not only for symptoms, but also to affect the biology of the disease. And a major obstacle is that diagnosis is confirmed only at autopsy. And I'll be highlighting the complexities of what we see pathologically under the microscope compared to what we see clinically. And this is a major obstacle for clinical trials where we want to target these specific pathologies in a living patient. If we don't have certainty in that diagnosis, that becomes a challenge. So we'll go through the clinical diagnostic criteria of FTD in detail, but I think what can be more informative in a way is to highlight a few vignettes to show the personal impact of this disease can have on individuals. And these are real patient stories to illustrate the core features of FTD, the behavioral variant, and their impact on caregivers. So this is the report from a mother in clinic saying that, recounting an episode where she fell and broke her hip, and she had to crawl into the car and drive herself to the ER because the patient didn't want to hurt his back to carry her or lift her into the car. And in this description, the patient was giggling, and not because they're a bad person. It's because of frontal lobe disease with lack of perspective taking. And this could be such a damaging symptom to family members, especially when the diagnosis is unclear. This could be an extreme psychiatric or perceived as a very extremely selfish behavior when in fact it's neurological. Another example is hyperorality and being driven to gorge on food and sometimes inedible objects. And this can be such an organic drive. We think it's due in part to damage to the hypothalamus and the orbital frontal cortex. But in situations, family members may have to lock or barricade access to food because of the extreme compulsion to eat. And there could be such intense gorging that patients may not have overt dysphagia, but could choke from overloading their mouth and have to have assistance for feeding to make sure that it's not excessive. And there is pica, eating inedible objects, as in the example here. Our frontal lobe and our ability to understand social cues is severely impaired in this disease. And particularly early in the disease, cognition may be mild or not affected to a measurable extent at all, but there could be severe deficits in our ability to understand motives and social cues from other people. And many of us often receive unsolicited scams online, over the phone, on TV. In this extreme example, a patient received an email to meet someone at an airport internationally and pick up a suitcase. And this ended up having illegal drugs in it. And the patient was incarcerated until it was determined eventually that the patient had FTD and was released. But extreme example to illustrate how vulnerable patients with FTD are, seemingly healthy physically often and cognitively, but having this impairment in judgment, which can and the ability to filter out potential dangers in society. So the personal health impact of this disease is enormous. I call the behavioral variant of FTD a tale of two brains, because the syndrome, while we have criteria and can accurately diagnose FTD compared to Alzheimer's disease, which is more of a memory problem, the underlying pathology that causes this syndrome is diverse, and I'll go through that. But in here in this box is the core clinical criteria for the behavioral variant. So we touched on some of these in the former slide, but behavioral disinhibition is a core feature that can include impulsive behaviors, going up to strangers, children, animals, we all have different social norms that can vary under different circumstances, but this is a clear departure from someone's baseline personality. Being gregarious could be a positive thing, but being too gregarious or volunteering personal information to someone we just met, that may be socially inappropriate. And the time of COVID, where social distancing is so important, patients with FTD may have difficulty standing in line at the cash register and want to move ahead, or be too close to not respect personal space. So behavioral disinhibition itself is heterogeneous. It could take all these different manifestations. Apathy is similarly can be less interest in personal hobbies and interests, which could resemble depression. But what's different than depression is it's not necessarily anhedonia, it's a decreased motivation that can take many different forms and could be extreme. There could be abulia or flattened affect, less motivation for self-care, for example. So loss of hygiene and wearing soiled clothing, for example, not being readily changing one's clothes, or a departure of someone's compartment in terms of dressing, bathing, and so forth. Ritualistic and perseverative behavior has several different features. This could be complex compulsions, where patients may have to do a certain activity repetitively, and if that gets disrupted, it causes agitation. It's different than in psychiatry, where OCD-like behaviors are relieving anxiety, and here there's no anxiety, it's more of a compulsion or a drive to do these behaviors. So for example, chewing gum every 15 minutes, having to change a piece of gum, having to walk on the sidewalk and avoid cracks, reading out street signs when driving down the road, and despite if there's a conversation going on in the car. There could also be simple motor perseverative behaviors, which more localizes to the frontal lobe. These are things like tapping, rubbing, humming, whistling, doing these kind of very repetitive behaviors. Loss of empathy, we talked about in the previous slide in an extreme example, but this could take the form of emotional detachment or coldness, so a loss of personal warmth, or overt disregard for other people's feelings. This is hard to measure in clinic and really requires, and should say for all of this, social cognition, we lack quantitative measures. So a detailed history with the patient and a reliable caregiver is very important. Because again, early in the disease, cognition may be minimally impaired, so difficult to detect and differentiate from psychiatry, for example. Hyperorality, we discussed, could take the form of gorging behavior, seeking carbohydrates, seeking cigarettes or alcohol, and even pica. And then the neuropsychological profile in general for FTD, or the behavioral variant, tends to be more executive difficulties, so trouble with planning and organizing, what we call set shifting, going from one task to the other, being perseverative or stuck on one task to the next, with relative preservation of episodic memory, such as list learning tasks, recalling words after a brief delay, or recalling elements of a drawn figure after a delay. That's more dependent on the hippocampus, which is more of a sign of Alzheimer's disease, and visual spatial functioning. So construction tasks like intersecting pentagons or the ray figure, they're usually preserved in FTD. So this constellation of symptoms are what we use to describe the clinical syndrome, and the clinical syndrome maps well to the imaging that's shown here in a sagittal view. You could see demarked by the red dashed line, what is a classic knife edge atrophy and severe frontoinsular anterior temporal loss of tissue that defines the syndrome. Here's a gross specimen of a patient with FTD, or behavioral variant, to see the striking focal cortical atrophy and the similar distribution. But even a neuropathologist can't fully diagnose FTD without microscopic examination. And when they do that, they have a different nomenclature called frontotemporal lobar degeneration. So pathologists and neurologists and other healthcare professionals who see patients, we speak a different language. We call this behavioral variant FTD. Pathologists call this frontotemporal lobar degeneration. And what you're seeing here these two different arrows, are the two main classes of pathology that are associated with the syndrome. And here in this box is color-coded to show you that roughly half of patients have tau, the tau protein aggregated here, while roughly the other half have TDP-43 here. The yellow line that's slim, that's about 5 to 10%, represents Alzheimer's plaques and tangles. And not to complicate things, but younger onset Alzheimer's patients who have the biology of Alzheimer's plaques and tangles, it's not uncommon to present with FTD syndromes and can present with a similar behavioral syndrome. But to focus on FTLD, or what's considered the frontotemporal lobar degeneration spectrum, we have these two very different pathologies. Not only do they appear different under the microscope, they're classified by different proteins that aggregate in brain cells. And we think that the aggregation of these proteins and the spread from cell to cell, or that propagation in a prion-like manner, is a key pathogenic feature of these diseases. So if we could detect these specific proteins, slow or stop their spread from neuron to glia to other neurons, we may be able to slow or stop the disease. The distinction between these proteins and a true bona fide prion, such as prion-related protein in Creutzfeldt-Jakob disease, is that we don't have any evidence that this could transmit between individuals, which is an interesting discussion. But to focus on the proteins themselves, these proteins have very different functions. So the tau protein is illustrated here on the left, is a microtubule associated protein. Its job is to help stabilize the skeleton of neurons and glia. And in the setting of tauopathies, including Alzheimer's disease, it becomes hyperphosphorylated, means it gets modified, it's unable to do its role in stabilizing the microtubules and starts to form tangles in neurons. And we have animal data and cell model data that shows that these tau aggregations can form seeds that could then spread from cell to cell. In contrast, TDP-43, the other main class of pathology that causes FTD, has a very different function and different location within the cell under normal conditions. So you can see here in the cartoon in red, it's normally found in the nucleus. Its job is mainly regulating gene expression, splicing of DNA and RNA. For reasons that are unclear, in the neurodegenerative process, it becomes trapped outside of the nucleus and aggregates in the soma, as you can see in the photomicrograph. And there's similar model data to suggest that TDP-43 aggregations could cause seeds that could spread from cell to cell. So if we want to develop drugs that could slow or stop this process or affect the normal function of these proteins that is lost, we need to be able to detect this in a living patient. And at this point, this syndrome gives us essentially the flip of a coin of one of these two very different diseases biologically. It's more complex, and I won't get into the details to illustrate here, though, but within these groups, there's very different morphologies or different subtypes of FTLD tau, or tauopathies, as we call them, neurodegenerative diseases that are characterized by the accumulation of tau versus neurodegenerative diseases that are characterized by the inclusion of TDP-43, including here you could see a lower motor neuron aggregating TDP-43 and ALS. So ALS, that we think of as a neuromuscular disease, which it is clinically, is within a spectrum of FTLD with TDP-43 and has very similar biology due to this accumulation. So how to parse this disease into biologically meaningful groups is still a matter of debate, but based on what I've outlined for you and some genetic data, there's strong rationale that a good starting point, at least, is that we could detect all the various forms of tauopathies and distinguish them from those with this very different protein aggregating in the brain, TDP-43. This would be much more likely to have the ability to test and develop meaningful therapies that could be specific for these very different biological disorders. This is an overview of the complexity of the syndromes for FTD. It's complex on purpose, and we don't have to go through all the details, but want to highlight a few important issues with the relationship between what we see under the microscope and what we see clinically. And the boxes represent the syndromes. We talked about the behavioral variant in the upper left. There are also language variants called primary progressive aphasia that affect language predominantly without prominent behavior or cognitive features. And there's two different types: the semantic variant, which is more loss of word knowledge, which is interesting and distinct from stroke aphasia, and then a non-fluent primary progressive aphasia that similar to a Broca's aphasia, where speech can have a motor speech impairment with dysarthria, trouble with grammatical expression, and production of fluent speech. And these are color-coded by the frequencies of pathology. Blue, how often do we see TDP-43 associated with these syndromes? Red, how often do we see the different tauopathies associated with these syndromes? And yellow, how often do we see Alzheimer's disease and some other less common pathologies in green and purple? And what you see is that the aphasia variants map quite well to anatomy and have some predictive value. About 70% of semantic variant is TDP-43, and similarly about 65 to 70% of NAPPA or the non-fluent is a tauopathy, but it's not perfect. And in considering a clinical trial, if you were going to enroll just these patients by syndrome alone, you'd be losing statistical power because you have patients with a different disease included. Cortical basal syndrome is very heterogeneous, as you could see here. 30 to 40% of patients with the frontotemporal syndrome, often lateralized Parkinsonian features that we call cortical basal syndrome, can have Alzheimer's pathology, but not uncommonly CBD tauopathy, which was the original name, but now that we know that that syndrome could be caused by many different pathologies, we call it cortical basal syndrome, or about 15% TDP-43. The motor syndromes, progressive supranuclear palsy, which we'll talk about in a little bit more detail later in the talk, and motor neuron disease or ALS, are much stronger predictive of a specific pathology. And they overlap. So all of these syndromes can converge over the course of a disease. And if we see signs of motor neuron disease or signs of progressive supranuclear palsy in these FTD cognitive syndromes and behavioral syndromes of FTD or primary progressive aphasia, that can help us identify the underlying pathology. But we really need biomarkers or new tests that can reflect the complex biology that goes with these syndromes. So the scientific question that I've been very interested in comes from seminal work from about 10 years ago. Bill Seeley from UCSF, this is a paper showing behavioral variant FTD patients MRIs, and the blue shows where the part of the brain that has atrophy. And you could see the knife edge atrophy pattern up top, where we see tissue loss, which we know is where the disease grossly affects the brain. And using an fMRI seeded network, which just means looking at blood flow on the functional MRI, we could see what parts of the brain are synchronized. And if you seed an area or look at the MRI data and say, in this area of atrophy, what other parts are synchronized or have similar blood flow, which we infer as connectivity, what you find is that the atrophy maps very well to a cortical network called the salience network that underlies our ability to do social cognition and executive functioning, so things that we use to define the syndrome of FTD. And he found this, and that group found this across all different neurodegenerative syndromes. So the clinical criteria that we use to define our syndromes maps well to cognitive networks and areas of the brain that are atrophic. Autopsy confirmed imaging data is rare, so we don't really know or understand where tau and TDP-43 fit into this system. And here is a map on the bottom from a more recent paper from that group showing MRI atrophy across the different tau and TDP pathologies associated with the behavioral variant. And what you see is that there's a lot of overlap. These different microscopic diseases, tau, TDP-43, converge in these gross anatomic regions and these networks of distributed connected regions that give us the syndrome of FTD. But I'm interested in also is how they diverge or how they're different. they have such different roles, these proteins. And if we can uncover these differences, that may tell us about the biology. And the overarching hypothesis in my lab is that despite this convergence on gross anatomical networks to give us the syndrome of FTD, the pathologies themselves have different cellular patterns. And if we can detect those, that can help us identify therapeutic targets, knowing the specific neurons that are vulnerable to tau preferentially versus TDP-43, for example. And we can use these differences to help design imaging, biofluid, or other biomarkers or new tests to help us be more certain that not only this is the behavioral variant of FTD, but it's due to either tau or TDP-43 pathology. To do this, we have a robust research program, and I lead the clinical core where we collect prospective natural history studies in our FTD patients. This is annual clinical exams, blood, DNA, spinal fluid, and imaging, including MRI, so we can understand the natural history is an important part of this to collect data that can help us understand these questions. And we do brain donation for research, but we also have a written report by the neuropathologist that I review with families so that they can get a final diagnosis of what the specific pathology that caused the syndrome. And that pathology data or that categorical diagnosis is important to validate what we've collected during life. So we could say spinal fluid from patients with tau differ in these measures compared to those who had TDP-43. And what my lab does then is takes the autopsy tissue and we try to measure the pathology more objectively. So not only do we have that final diagnosis, but we could start to measure the cellular features of tau, TDP-43, and connect it back to the syndrome and start to work towards a more precise diagnosis that matches the biology. To do this, we have two different ways of looking at histology. It's a collaborative effort, including colleagues from radiology and pathology, and I'll spare a lot of the details, but just to show that when we think of pathology as the gold standard, we look at a very small amount of tissue. So in the upper schematic, you could see what's called midfrontal cortex. We're used to looking at the equivalent of a fraction of the width of a piece of paper in a very small thumbnail-sized area of the brain. And standard histopathological sampling for diagnosis is really optimized for Alzheimer's disease, looking at the hippocampus and a few other regions. So maybe about 3 or 4 of these boxes are what most labs do for diagnosis. And what we've been doing is taking more regions that have importance for the social cognition that we described in language and looking across hemispheres. This disease is often lateralized to one hemisphere more than the other. And to complement that, taking these paired samples across hemispheres that are fixed in an identical way, we now are able to image a whole hemisphere and produce a custom mold for every brain where we can cut and precisely know anatomy and map it back to the MRI. And this lets us look in a very flexible way where we can get a survey of the whole hemisphere in these bigger size blocks in about 25 to 30 blocks per patient. And if we wanted to go into more detail of anatomy in a given region, we can cut through that block and do serial sections. And all of that tissue is preserved indefinitely. We've developed open source image analysis tools that helps us measure the pathology, including machine learning methods that rather than the standard but important neuropathologist eye quantifying, semi-quantitatively rating the severity of pathology, we want objective measures. And we have all of this data stored into a database and linked to all the clinical data we collect during life. So it's very powerful. So not only people like me who's interested in the pathology has experience looking at it under the microscope, but also colleagues who study neuropsychology or other aspects of FTD research can use the data for their studies as well. So that highlights our overall hypothesis. I want to touch briefly on some data that we have generated to start to model what's happening on a cellular level in these different pathological forms of FTD. So a few years ago, we looked at autopsy confirmed imaging. So that had white matter integrity, DTI data in a small group of patients. And we found that a long tract in the superior longitudinal fasciculus was preferentially vulnerable to tau. And we were able to look at the tissue of those same individuals that we had MRI during life, and in a semi-quantitative way, was able to rate the amount of white matter loss, as you can see in these in these photomicrographs here, where we see more tau inclusions in white matter and more loss of the blue myelin that correlated with the imaging. So promising that there's some biological differences in white matter. And more recently, we looked at this in our brain bank using those smaller tissue sections that I highlighted before, and using digital methods, as you could see here. So this approach lets us look at close to 100 brains in multiple regions. Is very high throughput, and you can see the area occupied is just how we quantify the pathology, and we account for all the different specific forms of tau and TDP-43. We use specialized statistical models that account for repeated measures and the inherent difficulty of missing data when we have archived tissue. Some of this tissue is from as long ago as the 1990s, where we may be missing a block, but we can still extract meaningful data from the whole data set. And in this study, we compare just how much inclusion burden is in tau and TDP-43 and white matter, because histopathological study of white matter is lacking in the literature. There's not much on it. And we found, similar to that early study, that overall the tau group had more pathology in the white matter than TDP-43. And when we looked at all the different subgroups, we found consistent results. So despite that heterogeneity within tau and within TDP-43, severity of white matter disease is somewhat of a unifying feature of the tauopathies that's distinct from TDP-43. We then looked at myelin. This is harder to quantify, and more recently we're working on metrics for this, but we found similar findings looking at the loss of white matter, as you could see here, a myelin stain, where a darker blue is healthier myelin and a lighter stain in the tauopathy groups was more likely. And this amount, mild, moderate, severe myelin loss, correlated well with the inclusion burden. So that was a good way to help validate our methods to detect the inclusions. And we did see some preferential difference in frontal regions for loss of tissue in tau and TDP-43. We can start to map the pathology back onto the brain, and this just shows the 5 main regions that we had in most of the patients. And the heat map shows the relative density of the protein burden. And in a data set this big of almost 100 patients, we can compare tau and TDP-43, the different proteins, but we can also compare the syndromes. Those patients with the same pathology but had primary progressive aphasia compared to those who had the behavioral variant had subtle differences in the distribution of their microscopic disease. So this suggests that both the clinical syndrome is important to localize where in the network the pathology is, but importantly, despite that clinical differences in the biology, tau pathology had much different distributions compared to TDP-43 on a cellular level. So if we can get that resolution and see this in a living patient, if we can detect measures of white matter, for example, we might be able to tell these diseases apart. We built on this to look at cortical layers, and I won't go through all the details, but we had similar statistics. We were able to look at the upper cortical layers, the deeper cortical layers, and white matter, and measure pathologic burden across all of our different pathologic subgroups. And we can use statistical models to help us understand how the pathology is affecting neurons and layers, because we know that upper layers have more connectivity in local circuits, where deeper layers have long-range projections and white matter, and that might help tell us, despite affecting those networks the same way, perhaps these different pathologies have microscopic circuits that differ, and we can start to detect those. So we sampled across all of our different brain regions that had different cortical layer structures, and we found in this layer ratio that's illustrated here, where a higher number means more pathology in the upper layers compared to the deeper layers, while a lower number was more pathology in the deeper layers than the upper layers. And we found that most of the tau pathology was greater in the deeper layers compared to TDP-43, which was more prominent in upper layers. So on a microscopic scale, looking at cortical layers, these diseases on a group level seem to differ. We looked across all the different clinical, genetic, pathological variants, and while there is heterogeneity like we observed in white matter, there's still overall, each individual tauopathy group had a different distribution across layers, including ALS, which we know has deep layer Beth cell involvement in the motor cortex, but overall still has greater relative TDP burden in upper layers, suggesting that TDP pathology may be affecting these interneurons, for example, that have more local projections. We then statistically modeled the relationship between layer pathology and white matter, and we were able to then hypothesize how we think the pathology is spreading. And in this example, you can see that TDP43 remains relatively constant in the upper layers without an association with white matter, whereas tauopathy is a more deeper layer pathology there is, the greater white matter pathology. And while we can't model this in a human brain and see in real time where tau is going because we don't have those imaging markers yet, this postmortem work suggests that tau may be influential. The spread through white matter may be an influential method of that disease process, which is different than TDP43. We have different ways of looking at tissue, and using the high resolution MRI, we were able to detect iron. And what you could see here from a recent series of brains that we looked at with that second process of tissue where we take a whole hemisphere, put it in a high resolution MRI overnight where we can get 160 micron resolution or close to microscopic. And we see dark areas that reflect iron, and iron overload is associated with inflammation and gliosis. And what we found is that the iron accumulation, at least in this initial series of patients, mapped the tau and TDP burden that we saw in the previous study, where upper layers of TDP43 patients had a dark band that corresponded to iron. When we looked at the cells involved, we saw iron-rich gliosis, compared to tauopathies had more of this irregular deep iron band that you could see here that related to different glial cells. So this suggests that different patterns of inflammation may be happening, and importantly, this layer difference may be something that can help inform us on how this disease spreads and advances so that we can stop it. To illustrate this in a different way, we're taking this work to start to develop what would be a virtual biopsy. So this is two regular MRIs in living patients who had the same syndrome of the behavioral variant FTD. In this case, both had more right temporal lobe disease as seen in the red box. The patient on the right was a little bit more advanced at the time of the imaging, but the overall anatomic pattern, cognitive network is identical. Clinically, they meet the same criteria, but when we have postmortem MRI, we can see these different patterns of layer iron. We see the deep irregular pattern of deep layer iron in patient one, whereas we could see the superficial upper banding of iron in patient two. And when we go to the pathology, it corresponds to the tau Pick's disease, tauopathy with the deep layer iron, and TDP43 with the upper layer iron. So this is suggestive if we can get imaging in MR that has sufficient resolution, we might be able to start to detect these pathologies in vivo is what we really need. So I'm going to skip through this part. I was just showing some more computational methods that we do with all of this data to help model the disease spread. But to summarize, our approach with digital pathology helps us model the disease propagation. We find these different cellular patterns of tau and TDP43 that implicate that despite having the same syndrome, there may be different mechanisms for it to spread throughout the disease, throughout the brain, particularly in cortical layers and long white matter tracts that may be more vulnerable to tau. And we need to develop more digital measures that can help us detect gray matter and white matter integrity directly. We want to start to involve spatial resolved gene expression data to help characterize the specific neurons that are vulnerable, and start to integrate the imaging that we collected postmortem with that during life. So in the last few minutes, I just wanted to talk about the translation and where we are now as a field in clinical trials for FTD, because this is an area of great growth and something that I did not foresee in my career as a cognitive neurologist studying neurodegenerative disease. And it's an exciting time. Clinical trials right now are focused mostly on the rare patients that have syndromes that are predictive of pathology, such as the progressive supranuclear palsy or classic Richardson syndrome, where the axial rigidity, supranuclear gaze palsy, and postural instability is highly predictive of tau. or the specific genetic forms of FTD, and most notably progranulin, which I'll talk about next. These patients, we know a molecular source that we can not one, have a diagnosis, but also have a mechanism to target with therapeutics. So progranulin associated with FTLD is relatively common. It's about 5% of all FTD patients, but 20% of those with familial FTD are those who have a positive family history. In our experience, familial FTD is about 20% of all FTD patients that we see. So much different than Alzheimer's disease, where a familial autosomal dominant inheritance pattern is relatively uncommon, about 5%. About a fifth of all FTD does associate with different pathogenic mutations, including progranulin as an important cause of that. These mutations tend to be autosomal dominant, again, so it doesn't skip a generation, highly penetrant. Most individuals who possess this gene pathology or mutation are likely to get disease during the course of their lifetime. And it results in haploinsufficiency, meaning these different mutations that are pathogenic amount to a roughly 50% reduction in granulin protein levels in the brain, as well as biofluids that we can measure. It's associated with TDP-43 type A pathology, so not to go through all the nuance, but there's a specific morphology of TDP-43, as you can see here, with these intranuclear inclusions. They call these cat eye inclusions. This pathological finding tends to map well to the progranulin mutation. This is from a patient of mine who had cortical basal syndrome. One that we talked about before was very heterogeneous, but often linked to tauopathies. At postmortem, seeing the TDP-43 made us be suspicious for progranulin. We were able to test the autopsy tissue and confirm and inform the family. So genetic testing is really an important part of FTD clinical care, because family planning opportunities are here now for clinical care, and trials are starting to be present for these specific molecular forms. So we have a robust genetic counseling program to help inform families who may be at risk and counsel them on the pros and cons of getting testing. FTD with progranulin has different clinical features. They tend not to fit cleanly into the diagnostic categories that I showed you before. They have a younger age of onset. They have a similar disease duration to other FTD patients of about 7 years, with a wide variability. They often can have motor features, Parkinsonism that's atypical, features of cortical basal syndrome. They can have aphasia, but often not the syndromes of just the semantic or just NAPPA, the non-fluent, but features of both or mixed features. So these clinical features are certainly not diagnostic of progranulin, but in the right context may help identify who may benefit from genetic testing. And genetic testing for all patients with FTD is, for those families who are interested, is not unreasonable. So because of the unique nature of progranulin associated FTD being a secreted growth factor that has haploinsufficiency or has less availability in the brain and in the body, it opens up different avenues to potentially treat. And in this schematic, you can see 3 potential ways that this can be done. One is through passive immunotherapy, and there's a current phase 3 symptomatic trial, which is ongoing now. We have it at Penn, and it's an international trial. And sortilin is a protein that breaks down progranulin. So by blocking this with passive immunotherapy, this could boost or do progranulin restorative therapy. And what's even more interesting about this trial is that it's also including pre-symptomatic individuals. So not only patients who have mild FTD, but also family members who have genetic confirmed mutation, but don't have symptoms, with the caveat that they have to have an elevated neurofilament light blood test. And the reason for that is because the age of onset is very hard to predict with familial FTD, and elevated neurofilament levels in the blood have been linked with what we call phenoconversion, or a patient going from no symptoms who possesses a pathogenic gene to developing FTD. So by enriching that cohort with individuals who are at risk of progression makes this trial more likely to see a biological effect and a clinical effect. So this is an ongoing trial for one mechanism for progranulin restoration. Another is protein replacement therapy by taking advantage of the transferrin receptor. That's a mechanism to cross the blood-brain barrier and linking progranulin protein to that. This could be passively infused and transmitted across the blood-brain barrier to boost progranulin levels in the brain. And then finally, gene therapy, which is done through an injection into the cisterna magna, and the early phase 1/phase 2 trials that are two different trials looking at different viral vectors, AAV9, AAV1. There's subtle differences between those and their relative tropism for neurons and brain cells, but both have a healthy copy of the progranulin gene. And if this gene can be successfully administered to the brain and be taken up by brain cells and secreted into the brain, there's good biologic rationale for all three of these mechanisms that by restoring normal levels of progranulin, we can slow or stop the neurodegenerative process, the TDP-43 accumulation. And this is ongoing now across many centers. We've dosed the first patient in the uplifted trial, and we've done 6 of the 11 patients in the proclaim trial. And data for those trials will be released by the companies, probably in the coming years or so. I want to briefly, to switch gears to PSP and just mention that the Richardson syndrome that we think of as highly predictive of tau, but the same PSP tauopathy, as you can see here, can be found in different amounts in red in different other FTD syndromes, including non-fluent primary progressive aphasia. So these patients are often excluded from trials because we can't be as certain as we are when we see the syndrome of PSP. And there's new clinical criteria for PSP that helps us detect the more subtle signs of PSP and these other FTD syndromes. And I won't go through all the details, but we take the 4 core features of the classic Richardson syndrome and use different levels of certainty, where, for example, the classic supranuclear gaze palsy would be the highest certainty, whereas slower saccades in the vertical plane or square wave jerks with eyelid opening apraxia could be more subtle signs. We could put together these different constellation of symptoms of different severity and have different criteria. So probable PSP would be good for a clinical trial where we want to target tau, but for epidemiology to detect more PSP in the context of behavioral variant FTD, non-fluent primary progressive aphasia, possible or what we call oligo PSP, these subtle signs may be helpful for us. So these criteria are ongoing validation, but I just want to highlight to end recent work that was done by Eddie Lee here at Penn, neuropathologist, doing postmortem work with 3 of our patients who participated in a recent immunotherapy trial against tau. So this was a trial that did passive immunotherapy for humanized monoclonal antibodies specific for tau to try to block the tau aggregation in the glia and neurons. And unfortunately, that trial did not meet its clinical endpoints. But when we looked at the brain tissue of these individuals, the type of tau pathology was different. Here you can see a non-immunized PSP patient has a class of tufted astrocyte. What Eddie found was that there were these punctate perivascular astrocytes, accumulations of tau. So the tau was not removed from the brain, unfortunately, but the morphology and markers of autophagy and lysosomal function were upregulated, suggesting that there was an element of target engagement. And although this is not where we want it to be, we want to see clinical endpoints. I think that there is good biologic rationale that hope can be renewed for FTD therapies, that we have these innovative ways that we can start to affect the biology, at least for the select genetic and PSP. And as we get more biomarkers and we can detect the pathology more accurately in living patients, these therapies will be more and more common. And there's significant parallels to Alzheimer's disease, where about 20 years ago, the first generation of immunotherapy for Alzheimer's disease, active immunotherapy for A beta peptides, unfortunately failed. and there were side effects of encephalitis. But as you can see here, the first autopsy report showed a reduction in change in plaque distribution in the brain of an individual who was immunized compared to what we see at plaque in an individual who is was not immunized with Alzheimer's disease. And here we are 20 years later with, despite there is some controversy, there's a lot of promise for amyloid-directed therapies in Alzheimer's disease. So the post-mortem work, I think, is an important piece to all of this to get towards therapies for FTD and related disorders. And I'll end now for questions and acknowledge wonderful collaborators that I had the pleasure of working with to do the work that I showed you today, and most importantly, patients and families who do the research with us, including brain donation, which this work would be impossible without their deeply meaningful contributions. So, thank you.
Dr. Irwin, thanks very much. It was a great talk, and actually, I think you really reviewed it nicely and showed the complexity and how heterogeneous this disease is. Because everybody, in a clinical world, if you're just seeing patients, you're just thinking about frontotemporal dementia, but in reality, I mean, that's probably the first question. After reviewing all this pathology, is it still fair to call it as a one disease? On a clinical basis. Would you? I mean, do you think we need to start moving away from the name frontotemporal dementia and start incorporating into the name the molecular findings or should we still stick to the frontotemporal dementia?
Oh, thanks, Peter. That's a great question. And it's an area of debate. There is a nomenclature group at NIH that is addressing this more broadly across Alzheimer's disease related disorders, including FTD. And one of the ideas that's been put forward is that trying to move away from a clinical pathological diagnosis like Alzheimer's disease, the word, where some of us hear that and think of a multi-domain amnestic syndrome. Some of us think of plaques and tangles, but they don't line up very well. In FTD, but our clinical labels do help us with communicating to patients the symptoms. So primary progressive aphasia, for example, is helpful because we could talk to patients about language and communication. And so, I think that decoupling the clinical from the pathology with one term, but instead using two different terms that are linked together. So for example, behavioral variant of FTD due to tau pathology or due to PIX tau pathology, depending on how precise our biological labels. So I think as we get biomarkers or tests, very similar, I think, to cancer biology, where there are syndromes that are associated with different tumor syndromes, but when we get the tissue, then we can link it to a specific molecular diagnosis. And I think that might be the best path forward, but it is complex, and communicating this to patients is an important piece of this too. So I think preserving those clinical syndromes and the importance for what that means for supportive care, but also starting to incorporate the biology into the diagnosis is important too.
Sure. Now, another question is about selective vulnerability. Why do these specific pathologists like those areas? Do we know that? What is so specific about, like, I don't know, salience network area, that starts affecting, cause the pathology? Are these because, I don't know, monoconema neurons are more, vulnerable, or there is, something else going on there?
Yeah, I mean, it's a really interesting topic. And right, there are some cells like the monoconema neurons that most of these pathologies do converge on that associate with the syndrome. And I think that there are some, we hypothesize, I don't have all of the data yet, but we're starting to stain for different neurofilament antibodies that characterize neurons. So for example, SMI32 is just a histopathological stain for large projection neurons, and trying to look at brain regions and SMI32 rich neurons that have long axons that theoretically, I would imagine, would be more vulnerable to tau because of the role of tau in the cytoskeleton. But that's an area that we really need to do a lot of work on. And I think some of the newer gene expression technology that's available now, where we can start to characterize neurons based on their gene expression and then look for patterns. The neurons that accumulate tau versus the neurons that accumulate TDP43, but also look at those who have behavioral variant compared to those who have primary progressive aphasia. So kind of looking at it by a 2x2, because there is there may be neurons that are specific for the syndrome, but also neurons that are specific for the pathology.
It's also interesting as that even, the genetic mutation, that runs in the family. One family member can present, it starts with it within the language network, and for another family member, it starts in some sort of, the social networks or in motor networks. What really determines that vulnerability? What is that? And that, I think, it's still a question unanswered. Another question is, how frequently do you see temporal limbic impairment in frontotemporal dementia, specifically in the tau or TDP43 pathology? Do you clinically, because classically frontotemporal dementia spares retention of the memory, but there are cases when it doesn't. So how frequently does that happen, and what determines that? Is that the secondary pathology or the primary pathology that just affects the temporal limbic region?
Yeah, that's a great question. So, probably about 20% or so the patients we see have a dense amnestic deficit, where they have problems with both visual and verbal memory. Particularly the semantic primary progressive aphasia patients with the focal anterior temporal lobe disease, we do see some memory loss with them, as others do as well, particularly for verbal memory. But you're right, it's not the most common feature, but it's not rare either. It's, so that while that behavioral variant syndrome, part of it is the relative preservation of memory, if you have all the other pieces, it doesn't necessarily exclude FTD. And then you brought up the very important point too of co-pathology. So as a young onset dementia, we see relative less co-pathology, but it's not uncommon, particularly in those older patients, where plaques and tangles of aging in the spectrum of Alzheimer's can coexist with tau and TDP43. And we do see those patients more likely to have more of a memory component. So co-pathology is another, complex problem with neurodegenerative research, is that usually, especially with older patients, multiple pathologies are there, but and that it affects memory function.
Another question, and I think I'm going to finish with this question, is about neurofilament light chains. One thing on the clinical basis, can we get those done? Can we, do insurance pay for that, for those tests? What would be the indications? And especially in frontotemporal dementia, I think the indication is clear, because frequently you don't, especially initially, you don't know whether you're dealing with a primary psychiatric disease or you're dealing with a frontotemporal dementia. And I think that could give you at least some good standing on where you're dealing. But is it easy to get that? And is the blood work as sensitive and specific as CSF?
Yeah, so this is great, and it's an area that's really exploding, not only in the context of trial eligibility and trial endpoints, but also for diagnosis. Like you say, if you have an individual who has a social presentation and we don't have clear progression to differentiate from psychiatry, an elevated neurofilament level in the right context could be strongly predictive of FTD. And even though it's not specific for FTD biology, we see it elevated with stroke, with MS, and so forth. In the right context, it can be very useful. Right now, it's still in the research realm, and those assays are getting more robust, where I think likely it probably will be available in the clinical space within the coming years. I know that the MS field has a strong interest in this as well, because apparently that's helpful for some of their responses to immune modulating therapies. And the work done in paired samples, so plasma sample and CSF, show a really high correlation. So that's another great advantage. As you say, lumbar punctures are somewhat invasive, but a plasma blood test would be, wonderful, very non-invasive. So I think this is coming. And it's really assay standardization and getting, a company to lead that forward and get that, FDA approval for clinical use.
Dr. Irwin, thank you very much. It was a great talk and very informative. And I'm getting all the responses from people, thanking for talk. I hope we'll be seeing you in future sometime again. And for all the listeners, thank you for joining. I would like to wish everybody a happy holiday season, and then we're going to connect again in the new year, 2023. Everybody have a good day.
Thank you so much. Take care.